Everyone to today's seminar on the power of stem cells. We look forward to this event every year where we have the pleasure of learning from Dr. Nitika Parmar and her amazing group of MS Biotech students on the latest stem cell research. Today, I get to introduce Dr. Parmar, who received her doctorate in molecular biology from UCLA and completed a postdoctoral fellowship at UCLA. She received her BS in biophysics and her MS in biotechnology and MTech in biochemical engineering and biotechnology from the Indian Institute of Technology, New Delhi. Nitika's fields of interest include molecular biology, genetic engineering, and DNA recombinant technology. Just a quick plug for our Ollie Summer Taste Program. It's wonderful to see so many familiar names in here. Current Ollie members, for those of you who aren't familiar, the Osher Lifelong Learning Institute is designed for adults 50 and above who want to continue the joy of learning. So we offer educational opportunities, no tests, no grades, just the joy of intellectual engagement and social connection. We have our Summer Taste program starting on Monday, which is a great time for new folks and longtime members to get involved. It's a series of one-time 2-hour classes, so you get a true taste of what Ollie has to offer. Registration is open and available on our website. And again, classes start on Monday, so great time to dive in. I won't take up any further time. I pass it to Nitika to start our seminar. Enjoy.
Excellent. Well, good afternoon, everyone. And thanks, Shaina, for the gracious introduction. It's always a pleasure for me to be with the Ollie students, so to speak. And I've been associated with Ollie for the past almost 17 or 18 years, having taught a variety of classes. And I've been doing this stem cell seminar for almost 9 or 10 years now with my students. And along with me are my wonderful 13 students who are currently in the stem cell embassies within the MS Biotech program. So for the first 45 minutes or so, I will be talking to you about stem cells, what they are and what their potential is. And then my students will take over. They are divided into 3 groups, and each group will be focusing on how a specific disease has the capacity to be addressed by the usage of stem cells. And those are pretty promising diseases to be treated here. And I would request all Ollie members to please save your questions till the end of my talk, and I'll be more than happy to answer those questions. So without further ado, I'm actually going to get started. Please bear with me as I'm sharing my screen. All right, I hope everyone can see my screen. Can someone just let me know if the screen is visible?
Yep, looks good.
Perfect. Thank you so much. So I'm just going to start very basic here about stem cells. As I mentioned, stem cells is a really big golden word for the past 20 years. So without diving into the glamour, the first thing we need to understand is what exactly stem cells are, why is there such a big hype about them, and what can they do in terms of fixing human diseases. So I'll keep my talk a bit basic, and I'll expect questions towards the end. So I want to open up with 2 Time magazine covers, which came up in 2001 as well as 2009. The 2001 cover was focusing on Dr. James Thomson, and he's credited as the man who brought us stem cells. And then about 8 years later, the Time magazine had a cover how stem cells were revolutionizing healthcare. And there was mention of 3 diseases on the cover: diabetes, heart disease, Parkinson's, and so forth. So naturally, for the past 20 or 24 years, stem cells have garnered a lot of interest in terms of how these can actually be utilized for fixing diseases. So just to talk a little bit about the key people, I really want to talk about very instrumental people who were responsible for the discovery of stem cells. The big credit actually goes to Dr. James Thomson at the University of Wisconsin-Madison. He was the very first person who was able to isolate embryonic stem cells And embryonic stem cells are commonly referred to as ES cells. And you can know by the name, obviously, they're going to be derived from embryo. In 1995, Dr. Thompson derived embryonic stem cells from monkeys. And in 1998, just 3 years later, he was the first person who isolated human embryonic stem cells from leftover embryos collected from in vitro fertilization clinics. These were frozen embryos. And then in 2007, Dr. Thompson made a revolutionary discovery. He developed a technique for transforming adult skin cells back into an embryonic stage, which are referred to as induced pluripotent stem cell, which I'll talk about a bit more later. So Dr. James Thompson is the really big person, and without his discoveries, we would not actually have much information about embryonic stem cells. And this was followed by two separate scientists, Dr. Gurdon from UK and Dr. Shinya Yamanaka from Kyoto University in Tokyo. And combined, they shared the 2012 Nobel Prize in Physiology of Medicine. So what did they do that they discovered that they received the Nobel Prize? Dr. Gurdon was actually responsible for reprogramming of frog cells. This was a very interesting experiment where the nucleus of an adult frog cell was taken out and put into an embryonic, or rather an ova, where the nucleus of the egg was removed. And he was able to actually show that an entire frog can be formed through that particular transfer. And in 2006, this is a very big name in stem cell sciences, Shinya Yamanaka, he did a revolutionary experiment where he took adult mature cells, which are fully differentiated, and he was able to revert them back into time by introducing a set of 4 different DNA fragments, and we call them genes. And these cells, which were initially mature and differentiated, which are basically adult cells coming from the body, they were reverted back into an embryonic stage. And these cells were now appropriately labeled iPSCs. And iPSC stands for induced pluripotent stem cells. And as a result of this discovery, there was no need to actually work with human embryos, which have their own aspect of controversy and ethical issues. So all these 3 scientists, Dr. Thompson, Dr. Gurdon, and Dr. Yamanaka, are probably the biggest names today in stem cell research, and they have been appropriately honored through a variety of prizes here. So what exactly are stem cells? So when we talk about stem cells, the first reaction I get from people who are not scientists are embryonic stem cells. So there's no much information about adult stem cells. And the first question comes up with, oh, we have to destroy embryos. We have to kill life. We have to, jeopardize embryos to get the stem cells. And stem cells are harmful because we have to destroy life. So the whole question is, are stem cells a boon or are they a bane? That's something that I hope you will answer by the end of my talk. So let's start with the basic definition of the word stem. The stem simply refers to a fundamental, simple progenitor line from which all other cells arise. So this is the basic, old, original cells through which all other cells in the body arise, whether it's neuronal cells, liver cells, kidney cells, you name it. That's the original cell, which is known as the stem cells. So what exactly is a stem cell? So for a cell to be classified as a stem cell, it absolutely needs to have 2 very specific features. The first one is it should be able to divide and make more copies of itself. And that is known as the capacity for self-renewal. It can make millions of copies of itself. And so it can lead to the formation of daughter cells. The second very interesting capacity of a stem cell is differentiation. where it is specialized to give rise to different kinds of cells within the body: a muscle cell, a blood cell, a nerve cell, a liver cell. So stem cells have to have both the features to be able to be categorized as a stem cell. There are some cells in the body which can differentiate, but then they don't go back in time and they don't divide further. So those are not stem cells. And there are some cells in the body which can make several copies of themselves, but they cannot differentiate. So those are also not stem cells. So if a cell has these two features, it is classified as a stem cell. So stem cells, as I mentioned, are initially undifferentiated. So what do we mean by the word undifferentiated? So undifferentiated means it has not acquired very specific features which dictate the cell to go towards a specific lineage. That means they either express certain genes or they turn off the expression of certain genes. Whereas stem cells in the initial stages are pretty progenitor, pretty primitive. They are unspecialized. But they have a capacity of dividing sort of indefinitely. So that is known as self-renewal. For us as biologists, the second property is very important. That means the potency. How potent are these stem cells to actually generate different kinds of cells in the body? And this potency is dictated by a variety of environmental conditions, by experimental conditions in the lab. So these cells are pretty sensitive. Depending on what kind of food you give to them, when I say food, I'm talking about different growth factors. I'm talking about different genes that can be turned on or turned off. So they're pretty sensitive. And depending on what food we give them or what genes we turn on or turn off, these cells can be forced to become certain specific cell types in the body, like I mentioned, kidney or neuron and so forth. So this ability, the potency, can be capitalized within the lab in a petri dish, in a cell culture house, to be able to study how stem cells can be developed or differentiated into different types of cells within the body. And this is extremely important for developmental biologists to understand how an embryo actually grows in response to different gene triggers. So self-renewal means it simply is responsible for maintaining the population of the cells. So stem cells are constantly maintaining their own kind. So one would ask, why are stem cells dividing? And why do they replicate to make more copies of themselves? And the answer is very clear. We can see we are doing so many different things. The human body is always working 24/7. The heart is beating, the neurons are conducting their signals, the muscles are contracting and relaxing. So there's always tissue repair which is happening in the body. So when the cells get tired, they actually kind of die off. So in order to replace those cells, that's where the stem cells come into play for tissue renewal. Otherwise, if the cells become old and they die off and there's no replacement, the human body is likely to die. But these stem cells are great. Whenever there is a need for tissue repair, the stem cells start renewing themselves, and then they can differentiate to replace the different cells that have died naturally within the cell. So both these features, renewal and differentiation, is very important for maintaining the health of the body, for replacing damaged cells also. So I'm going to talk about the word potency here, because I used this word initially, that stem cells are highly potent in giving rise to different kinds of cells. So what do we mean by the word potency? The potency can be classified into three different kinds: pluripotency, multipotency, and unipotency. We'll start backwards here. Unipotency means a stem cell can produce only one cell type. So if we have a stem cell which is sitting in the brain, that can differentiate only into a brain stem cell. So that potency is referred to as unipotent. Multipotent means one stem cell can give rise to a variety of cells, but within the same family. So for instance, the classic example is the blood cell. The blood cell can differentiate to give rise to red blood cell, white blood cells, platelets, basophils, neutrophils, eosinophils. So a multipotent stem cell can give rise to cells within the same family. And the gold standard here is the last one, which is pluripotent. Pluripotent is also referred to as totipotent, and such a stem cell can differentiate into any cell type within the body. You name it, it can differentiate into a hair cell, it can differentiate into a kidney cell, a pancreatic cell, and so forth. So, of all the cells within the body, there is only one cell type which can be classified as pluripotent, and that cell is embryonic stem cell. There is no other cell in the body which has pluripotency capacity. An embryonic stem cell is found in the embryos, in the fetus, essentially, as the fetus is growing in the very early stages of the embryo. Otherwise, blood stem cells are multipotent. Other organ stem cells coming from the liver or the kidney are unipotent. So naturally, for scientists, particularly those who want to study the development of organs, the pluripotency aspect is very important. And so the utilization of embryonic stem cells is actually very valuable. But then again, the controversy comes into play. Well, we need to destroy the embryos. And now we have come a long way where we have moved away from the utilization of embryonic stem cells as a result of the discovery of induced pluripotent stem cells that I had mentioned earlier, which I'll talk about in just a few slides. So what are the various types of stem cells in the body? There are four predominant kinds. The first one is embryonic stem cells, referred to as ESCs. The second is adult stem cells, and naturally adult means they're coming from adult differentiated tissues. The third is coming from the placenta, cord blood cells. This is the tube which joins the mother to the growing fetus, which is providing the nutrition to the growing baby in the womb. And the fourth kind is the amniotic fluid-derived stem cells, also known as AFS. And the amniotic fluid is the fluid which is bathing the growing fetus within the womb of a mother, and it provides a cushion as well as nutrition to the growing fetus. And this amniotic fluid is actually filled with certain populations of stem cells also. So where are embryonic stem cells coming from? So the start is the fusion between a sperm of a male and the egg cell of a female. So once they fuse, whether it's done through a sexual interaction naturally, or it is done in the lab by artificial means, by taking a sperm and mixing it with an egg cell and waiting for 2 or 3 days to watch the development of what we call the zygote, that is an embryo. And once that zygote or embryo reaches about 4 or 5 days, now that embryo is referred to as a blastocyst. So you don't need to really worry about the scientific terms here, but I'm just going to show you from where are these embryonic stem cells isolated. So in this figure, you can see 2 stages, an early stage of the growing embryo and a late stage. And the blue cells are referred to as the supportive cells, but the real important cells here are the orange cells, which are referred to as the inner cell mass or the ICM. And if anyone wants to isolate embryonic stem cells, this is where they are coming from. The orange cells are the cells which are isolated from embryos, and then they're grown in the lab, and those are referred to as embryonic stem cells. So needless to say, the embryo absolutely has to be destroyed if one has to isolate these orange cells. There is no question about it, because the embryologists have to go with a little bit of a needle. It's a very fine needle. You've got to poke the embryo and you suck out these orange cells. And in the bargain, the embryo actually loses its life here. So these are very interesting electron micrographs of different stages of the embryo as it grows. Here we have an early blastocyst, a late blastocyst, a 4-cell stage, and a modular stage. And you can see this is the portion, the inner cell mass, which is the origin of embryonic stem cells. So what are the challenges when we isolate embryonic stem cells? So what do we do with the embryonic stem cells once they are isolated? Well, the first thing we do is find a good recipe for feeding them. So embryonic stem cells, of course, now we know what food they need, but in the initial stages, there was a lot of hit and trial which was done to make them grow in the lab. And that is almost 24 years old now. So now we have fantastic recipes of growing and maintaining embryonic stem cells. So that's well and good. That's just self-renewal. But how do we have them differentiate into turning into skin cells or blood cells or liver cells? Now, these cells, which are shown on the right side, are extremely specialized cells. A skin cell cannot turn into a neuron. A blood cell cannot turn into a liver. All these cells in the body are highly differentiated. They have certain genes which are turned on and certain genes which are turned off. So let's say if there are 100 genes turned on in the skin cell, it's not necessary that all 100 are turned on the neurons. Maybe only 55 are turned on in the neurons, and maybe only 67 are turned on in the blood. So the point I'm trying to make here is it is not at all easy to convert an embryonic stem cell into all these different differentiated cells, cell types within the body. And it took years and years of hard work in terms of feeding the embryonic stem cells different cocktails of growth factors. Growth factors are basically different peptides and hormones, which are very nourishing for the embryonic stem cells. And different kinds of conditions will give rise to different differentiated cells in the body. So you can imagine for the past 18 or 20 years, scientists have been working with different kinds of foods and different kinds of peptides and hormones and growth factors to see what kind of cells are generated by feeding embryonic stem cells different kinds of cocktails. And now we do know what kind of genes need to be supplied to the embryonic stem cells to have them turn into skin or neurons or blood or liver or whatever different type of organ we're talking about here. So that's embryonic stem cells. The second type is the adult stem cells. And as is evident from the word here, adult stem cells are not isolated from the embryos. They are isolated from a fully grown, highly differentiated tissue or organ within the body. So if I were to take a liver, I would say 99.5% of the cells in the liver are highly specialized, highly differentiated, and performing the function of detoxification through the various enzymes within those cells. But 0.4 to 0.5% of the liver is comprised of a section of undifferentiated cells, which are referred to as adult stem cells. Very, very small percentage. But that small percentage is very important because when the need arises, those stem cells can rapidly divide and those function for maintaining tissues or getting rid of damaged cells and so forth. So now these adult stem cells are known to be very important for tissue repair, and they have been found in pretty much every organ in the body. You name it, muscle, skin, liver, bone marrow, brain, they are present everywhere. So this is just a picture of showing you where the adult stem cells are isolated from and where the embryonic stem cells are isolated from. And like I mentioned here, embryonic are totipotent or pluripotent, while adult stem cells tend to be multipotent in nature. So now, for the past 20 or 25 years, scientists have been able to show that pretty much every organ in the body has a very, very tiny population of stem cells. And those stem cells are very necessary for tissue repair. But there is a flip side to it also. If we get benefit from these adult stem cells, whenever there is a cancer or there is a tumor in the body, even that cancer or tumor has what we call CSCs, commonly known as cancer stem cells. And this is a big problem in cancer therapy. So even if we are able to remove 99.999% of the cancerous cells from the body, it is that 0.001% of cancer stem cells, which are very hard to target, because they are buried deep within the cancer and they're very hard to get rid of. If that 0.001% still remains and is not killed by chemotherapy or radiation or whatever therapy a person is undergoing, that cancer stem cell, because of its capacity for self renewal, will regenerate the cancer all over again. And that is why people who are deemed cancer free within a span of 15 or 20 years, it can actually relapse and the cancer can occur again. And this happens as a result of cancer stem cells. So I just wanted to mention this here because CSCs have become a very hot area, particularly the Institute City of Hope Beckman Center, which is actively working on cancer stem cells, trying to find out how do we kill the cancer stem cells so that the cancer is actually removed right from its root and there's no question of the cancer coming back here. So while adult stem cells are great, they are needed for tissue repair. On the flip side, the cancer stem cells are really bad because they will actually result in the reformation of the cancer. So, like I mentioned here, all these adult tissue stem cells, which can be found in the blood or brain, they are actually responsible for tissue repair, and those are very crucial within the body. So, the key question about adult stem cells is how many kinds exist? Of course, we have an answer today that majority of the organs within the human body have adult stem cells. And the big question is, what is their source? Are they left over from embryonic stem cells, becoming multipotent from pluripotent? So that is a big question. Are they arising in a separate form, or are they leftover cells coming from the embryo? And those questions are still being deciphered here. Another question is their plasticity. So what do we mean by the word plasticity? It's coming from the word plastic. So plastic is a fantastic chemical which has a capacity to be molded into any shape and form. Plastic can be made into a bowl, it can be made into a tube, it can be made into a block, you name it. And so in the same way, cells within the human body can be plastic. And embryonic stem cells have the highest plasticity. That means they can be turned into any cell type in the body. So adult stem cells don't have that level of plasticity. They can be plastic, but they can only turn into specific cell types. So a neuronal stem cells cannot turn into a blood cell and so forth. So that is why embryonic stem cells are preferred because of their high level of plasticity. And today, induced pluripotent stem cells are also deemed to be highly plastic. And the final big question for people studying tissue regeneration and regenerative medicine is the big question: what genes or what food or what factors or what chemicals stimulate these adult stem cells to sites of disease and injury? So when we have some kind of an injury in the body, which is very deep-seated, what kind of chemicals or signals are sent by that damaged tissue so that adult stem cells can come to the rescue and start dividing and start fixing the damage? So there's still a lot to be learned about adult stem cells because of all these questions, which are still being answered slowly by various scientists all over the world. Third kind is the stem cell, which is isolated from the umbilical cord. And most of these cells, majority of these cells are hematopoietic in nature. Hematopoietic means they are coming from the blood cell lineage, and they can only form new blood cells. That means they are multipotent in nature. However, some scientists in about past 15 or 20 years have done some very cool experiments, and they isolated the cord blood stem cells and they provided them the right food, which actually turned them into neuronal cells. But those neuronal cells do not survive too long within the cell culture environment here. But today, whenever a woman gives birth to a child, the doctors ask her whether she wants to preserve her placenta and she wants to preserve her cord blood stem cells. That is an option given to a woman here as she gives birth. And what is the reason for that? The reason is if the children of that particular woman have some kind of a genetic disorder later in life, which comes as a result of spontaneous mutation or for whatever reason, if the cord blood cells are preserved and they're normal without any genetic defect, those cells can be harvested and they can be utilized for repairing the damage in the subsequent children that the woman is giving rise to. And they come with a price. They're not free. The hospital actually charges a specific fee for isolating the cord blood stem cells from the umbilical cord, and then they're frozen down and kept in a freezer. And when the need arises, the couple may request for the utilization of those stem cells. And based on my conversation with some of my friends, a lot of women are actually uh, giving permission for the freezing down of their cord blood stem cells because they can come in handy much later, particularly when there is a genetic disease running in the family and you don't want subsequent children to be impacted by the same disease, in which case cord blood stem cells can be preserved. So this is just showing you an image of a placenta where a child is just recently born, and this placenta is then removed and the hematopoietic stem cells are isolated and frozen down and kept in these deep freezers for an indefinite period of time. And the last type of stem cells are known as AFS, which is amniotic fluid derived stem cells. And this is just an image which shows you the fluid, which is basically looking like lymphatic fluid, which is surrounding the growing fetus. And it's basically serving as a cushion to protect the fetus from trauma. And it also provides the fetus with nutritional factors here. So in order to isolate the stem cells, which by the way is not much, it's only 1% of the amniotic fluid, a skilled gynecologist has to go with a needle through the womb of the woman and then gently reach the amniotic fluid and make sure they're actually not touching the growing embryo, because if the needle touches the embryo is likely to die. So they have to be very, very specialized in taking the needle and just sucking the fluid from the amniotic fluid and then isolating the stem cells. But as of today, hardly any researchers are working with amniotic fluid stem cells because of the risk and the danger involved to the embryo. So majority of them today are working with adult stem cells as of today. All right. So now the question is, let's go back about 20, 25 years when embryonic stem cells were discovered. And I just want to let you know that even today, embryonic stem cells are completely valuable, particularly for developmental biologists. You want to isolate the inner cell mass, you want to grow them in vitro in little houses in the lab, and you want to monitor development, how the embryos develop, and what kind of genes, particularly at the molecular level, which genes are turned on, which genes are turned off, which is very, very important from a developmental point of view. For those cases, the embryos have to be obtained. So where do we get the embryos from? The number one source is, of course, fertility clinics, where individuals are doing in vitro fertilization trials where natural conception is difficult, and these individuals are resorting to fertilization between the sperm and the egg cell in a petri plate, which is referred to as in vitro fertilization. So whenever an embryologist is conducting an IVF trial, several eggs are isolated from the woman's body. It's just not one egg. In fact, the success rate is about 45 to 50%. So if 10 egg cells are used, only 4 or 5 will be successfully fertilized by a sperm. So if we take 10 egg cells and we throw sperm at them, and of these 10, maybe 5 are successful in growing into what we call an early stage embryo, and the couple says, we just want 1 or 2 kids, just 1 or 2, and 2 are implanted back inside the woman's body. What do you do with the remaining 3? Or if not 5, what if 10 embryos are left over? What do you do with those 10 embryos? Those are frozen down in these fancy freezers, and then the couple is asked after a few years whether they want to have more children or They don't want those embryos to grow further. So fertility clinics, even today, are actually loaded with leftover embryos because the couples have enough children and they don't want to have more children. So what do you do with these leftover embryos? These are not implanted back into a pregnant or rather into a woman, particularly after genetic screening. So what do we do with these embryos? Whether they are healthy or they have some kind of a genetic defect, they cannot be frozen forever because running these freezers is actually pretty expensive. They're very electricity consuming because they are frozen at minus 80 and minus 196. So these are pretty expensive. So fertility clinics are the source of embryos, particularly when 20 years ago embryonic stem cells were worked with. And this is just one of the mentions, Jones Institute in Virginia, which was providing the leftover embryos here. So why not use adult stem cells? Well, the distinction is very clear. Adult stem cells are not pluripotent. They're not as plastic as embryonic stem cells. So for embryologists, it's very important that they study embryonic stem cells if they want to study the entire cycle of differentiation. And another reason is that the existing stem cell lines have limited genetic diversity. So adult stem cells are less diverse compared to embryonic stem cells because they are multipotent versus embryonic stem cells, which are pluripotent. So the question here, this is the big can of worms that we open. We have both proponents and opponents of the utilization of embryos. So the proponents say there's hundreds and thousands of frozen embryos sitting in fertility clinics all across the nation, all across the world. So if the couples don't want any more children, these embryos have to be destroyed eventually. You cannot preserve them for hundreds of years because if you don't want them, why are they sitting in the freezers? So the proponents say, if they're going to be destroyed, why not use them for scientific advancement so that the cells can be isolated and they can be studied for how they can fix diseases? So that's where the science comes in. But the opponents say that the embryos are live, which is correct. An embryo is full life. And once it's implanted into a woman's body, it can generate a full child. So the opponents say the embryos are live and they should not be destroyed. They should be given for adoption. So both angles are correct. The science can advance by utilization of these pluripotent stem cells, while on the opposite side, from an ethical point of view, is destruction of life validated here. So what are the uses of these stem cells in the lab? A lot of uses, actually. We can study how development occurs at the cellular level and the molecular level. And very importantly, we can test new drugs and new therapies. We can expose these stem cells to different kinds of cocktails and see how the stem cells respond. And what is happening today is the development of cell-based therapies. So cell-based therapies means now we're utilizing cells which have been genetically engineered to express certain genes which were silent in a variety of diseases. And now when these cells are genetically altered, they are producing a protein which is absent in a disease. And you put these cells back into a patient. Now, because of the production of that new protein, those diseases can be fixed. And these cell-based therapies have become very popular now, although they are cost prohibitive. Even now, a single cell-based therapy can cost anywhere from $500,000 to $1 million for a single therapy here. And so not all of us can afford such therapies here. All right. So stem cells, one area where you probably know very well, most of you, is when somebody has blood cancer and they are subjected to chemotherapy or radiation, basically you're killing all the cells in the bone marrow because that's what the radiation is doing or the chemotherapy is doing. In blood cancer, you want to kill all the dirty blood cells. You want to get rid of all the cancerous cells. But then if the marrow loses all the blood cells, then the marrow is empty. So what do you need to do? So for cancer patients, in order for them to survive and regenerate the immunity and their blood cells, they have to undergo what we call stem cell transplantation. And this is routinely done now. This has been done for the past 25 or 30 years. So for transplantation purposes, the stem cells need to satisfy several criteria. They should be able to divide very rapidly. They should be able to generate quantities of tissues which can fill up the gap. They should be able to differentiate into different cell types. And most importantly, I think bullet number 4 is so important, that once they are transplanted into the human body, they should be able to survive. Not that you transplant them in the body, they're very happy and they're growing for 2 weeks and then suddenly they die off. And that is very detrimental to the patient. Once they are transplanted, they should be able to integrate into the surrounding tissue. They should be able to cross-talk with the cells already existing in the cell, and they should not harm the recipient in any way. So survival of the transplanted cell or the transplanted tissue is of utmost importance for transplantation success here. And of course, today, stem cells are utilized, have the capacity to be utilized extensively for a large variety of diseases. And some of the diseases which are listed here are cardiovascular diseases, autoimmune diseases, diabetes, and so forth. And the student groups will actually be talking about this very shortly. So moving away from embryonic stem cells, now we have moved into the era of induced pluripotent stem cells, which is referred to as iPSCs here. So this is basically giving rise to the progenitor embryonic-like stem cell coming from adult stem cells. So we can take fully functional, specialized cell types from any part of the body. Normally, we take the fibroblast from the skin and we feed them certain foods. When I say certain foods, it's a combination of 4 different genes or 4 different proteins, which are known as OSLM, OCT4, SOX, MYC, and one more. So there's 4 different proteins which can genetically reprogram these adult stem cells and take them back into time. So just imagine if somebody says, I'm going to give you some food, and you will go back and become a 10-year-old again. Of course, that's not possible. It's sci-fi. But that can be done in the lab, in vitro, where these adult cells are taken and they are reverted back to a very early stage. Although we don't call them embryonic, because they're not embryonic, but they are embryonic-like. And the new name which is given to them is induced pluripotent stem cells. And because of this discovery by Shinya Yamanaka, he actually got the Nobel Prize. And this is a stupendous discovery which has opened up a huge variety of scientific projects in the area of developmental and molecular biology. So these are just some images which show you how through genetic reprogramming, these adult stem cells can be taken back into time and be converted into pluripotent stem cells. And these pluripotent stem cells can now be differentiated into different cell types, whether it's for fixing diseases or for tissue repair and regeneration. All right. I'm just going to skip this slide here. So what are the controversies? I'm just going to end in the next 3 or 4 minutes. The first controversy was about the human embryonic stem cells. Again, it was a question of life and death. And the big agency on stem cell research has actually given this statement on its website. In the case of embryonic stem cell research, the end that scientists hope to achieve is the relief of human suffering. That this is a humanitarian and worthy end is not in dispute. The controversy is about the means, namely the consumption of donated embryos. Second ethical issue is with regard to the induced pluripotent stem cells. Now, induced pluripotent stem cells have to come from an adult. So the protection of the human subjects have to be ensured. Informed consent has to be there. Genetic identity has to be protected. And because of the immense power of the induced pluripotent stem cells, some rogue scientists can start conducting very unethical experiments by combining a human cell with an animal cell and trying to create so-called human animal hybrid stem cells. And that can be pretty detrimental. So that should be avoided at all costs. So there are, because of the immense power of these induced pluripotent stem cells, it is very important that there are ethical and legal guidelines for their biomedical usages. So that's another ethical issue. And the third ethical issue is tied to what we call somatic nuclear transfer. And basically, this is what I told you right in the beginning of the talk, where you can take an adult cell, you can take a cell from your skin, you can isolate the nucleus, and you take an egg cell from a woman, you remove the nucleus of the egg cell, and now you take the nucleus from the skin cell and you put it into the egg cell. And you transplant the egg or implant it into the woman, and that will give rise to a child. But now the nucleus is coming from an adult cell. It's not the natural nucleus of the egg cell, and it is not the nucleus coming from the fusion of the sperm and the egg. So in this case, through somatic nuclear transfer, one can generate children without the need of a male partner. Now that is huge, and that is referred to as reproductive cloning. So cloning can be of two types: somatic cloning, where the nucleus is taken and put into only somatic cells. So remember, there's two kinds of cells in the human body: somatic and germline. Germline are the ones which give rise to the sperm and the egg and eventually the children. But somatic stay within the body. They're not given down in terms of reproductive cloning here. So thankfully, reproductive cloning is banned in the world today. That means you cannot do somatic nuclear transfer to generate babies. But you can still do somatic nuclear transfer for fixing diseases, and that is very much allowed here. It's not prohibited. But reproductive cloning is strictly prohibited. Although just a few years ago, there was a rogue experiment done in terms of generation of twins who were genetically engineered so that they were resistant to infection by HIV. And that was reproductive cloning. And that completely bypassed all ethical regulations. And that scientist was banned for life here. I'm not going to talk about the name of the scientist here, but like I said, if this technology gets in the wrong hands, particularly making animal and human hybrids, that can take science in the wrong direction here. So, and this is just a picture showing you how somatic cell nuclear transfer is done. You take the nucleus from an adult cell and you put it into the egg cell, and now the egg cell is implanted into a woman. And then if it's allowed to grow to full term, it will actually generate a full child here. So scientists and stem cell researchers and agencies are doing their best for educating the public. They're having open dialogues now. They are doing clinical trials with great deal of caution. So in terms of education and in terms of funding, there's a lot of training which is being provided to investigators, to students, to staff members, to the public, and also to the lawmakers. So stem cell research today is highly, highly regulated, at least within the US. And the agency which is providing the funding in California is the California Institute for Regenerative Medicine, also known as CIRM, which was established as a result of the passage of Proposition 71, which was provided $3 billion for conducting stem cell research. And my 13 students who are presenting today are actually part of this program, the CIRM program, and some of them are receiving funding from this agency for conducting stem cell research in the internships, which are starting in just a few months here. So I'm just going to skip here because of the interest of time. I think my time's up, and I'm just going to end my talk here. And I am more than happy to take any questions at this point here. Does any of you, any of the individual in the public have any questions here? You can put it in the chat box, or you can actually speak up. Any questions? Yes, so there's a question on the chat. How long can cord blood be stored? Indefinitely. Years and years and years. It all depends on what temperature you're using for freezing them. So normally our refrigerators or freezers in the house are at 0 degrees. In our labs, we have three kinds of modalities for freezing different kinds of, you know, things like DNA or RNA or cells. There is minus 20 degrees, there is minus 80 degrees, and the coldest is what we call liquid nitrogen, which is minus 196 degrees. And you can just imagine nitrogen is a gas, but it is subjected to such cold that it actually becomes a vapor, like a liquid, and that is minus 196. Anything that is frozen at minus 196 can last for centuries. I'm going to look at the chat box here. Yeah, are stem cells working right now to cure any diseases? Yes, they are. In fact, the hype is around how do we fix diseases like diabetes? How do we fix Parkinson's, Alzheimer's? For that, we still have a few more years to go. But in case of arthritis of the knees, where stem cells have been injected to the knee pocket, that has shown to fix arthritis. In cases of blood cancers, stem cells have been shown to fix. And there's a couple of diseases where I believe when there's loss of sight or there is a rapid loss of sight, transplantation of stem cells directly into the eye, at least in the clinical trial, have shown that vision has been restored. So I would not say that 50 diseases have been cured, but through the various efforts of various scientists, we are actually getting to the point that in the next 30 or 40 years, several diseases will be cured. The other question is, doesn't liquid nitrogen temperatures destroy the cells? No. It all depends on how quickly you're freezing the cells. So normally we do not freeze the cells in the presence of water because these water molecules, they form crystals. And anytime you remove the cells from liquid nitrogen, these crystals, as they melt, crystals are basically like needles. They can poke into the cell membranes and they can destroy the cell. So your point is very well taken. But the cooling in liquid nitrogen is done so rapidly in the presence of a very specific chemical known as DMSO, but I won't go into it. And DMSO is referred to as a cryoprotectant. So it protects the cells. So if done correctly with the right cryopreserved fluid and also done with a great speed, the cells are not destroyed. And of course, once you thaw the cells, they have to be thawed in a very appropriate manner also. So there are scientific protocols of cryopreservation and also thawing of the cells. What is being done to regulate stem cell use globally? A very good question. Depends on which country we are talking about. In the US and most part of the Western world, it is regulated very tightly through the stem cell research agencies. But in some of the other countries where the regulation is not very tight, some scientists are taking on the onus of doing stem cell research, which technically should not be done. So I would say, at least I can speak for the US, where we are, there is very tight regulation here, at least. Global level, it's extremely variable, depending on what kind of resource we're talking about, what is the level of the education of the lawmakers, are they able to understand the power of the stem cells? So that is why I say, when we're talking about lawmakers, it's very, very important to have scientists as lawmakers, because science is best understood not by politicians, but by scientists. And that is why to inform how the regulation has to be done, we do need to have scientists on the board of directors, in terms of council members who are doing advisory roles so that the regulation is done pretty tightly. Question is, do you need your own stem cells for injection or can you inject somebody else's stem cells into your blood? Yes. So there's two kinds of transplantation. There is autologous and there is allogenic. Autologous means the stem cells are coming from my own body. So I'm taking my skin cells, I donate my skin cells, somebody grows them in the lab, somebody does some genetic engineering, they put some DNA in it. Now my skin cells are expressing a new protein, and that is a protein I'm missing in my body. So after those cells have been genetically engineered and they're producing the happy protein that I need in my body to survive, those cells are put back into my body. So that is known as autologous, in which case there is no rejection because those are my own stem cells. Allogenic means I am utilizing somebody else's stem cells. It could be my sibling, it could be somebody who has a perfect blood match, HLA matching, MHC matching, whatever it is. We are matching all these proteins on our membranes of the cells so that my body doesn't reject them. So both are possible, autologous transplant or injection or allogenic. When we do allogenic, it's very important to make sure that my body does not reject the foreign cells that are being injected into my body. So we do what we call HLA type matching, we do MHC matching, and so on. So there are various tools to ensure there's no rejection. How many modified stem cells are needed to allow the body to make repairs? That is a good question. Honestly, I don't know. I don't know whether we need 5,000 or we need 50,000 or we need 5 million. I actually don't know the answer to that. I wish I did. I can find the answer for you, but on top of my head, I really don't know. But considering the fact that they divide so fast, self-renewal, so if we start with, let's say, 50 cells, within a span of 24 hours, you may end up with 5,000 cells, and another 24 hours, 10,000 cells. So again, I'm just guessworking here. Maybe you don't need as many stem cells because of the power for self-renewal. So I think I'll stop here because I want to give you a 5-minute break before the students actually start their presentations. There's just one more question. I'm going to read it. If a birth mother doesn't want to save the cord blood, does it get automatically saved off? No, it doesn't get saved off because it is a tissue which is coming from the birth mother and that tissue belongs to her. Now, if a scientist comes to the mother and says, I need your placenta because I do umbilical cord research, can I get your placenta? So if she says yes, and she gives her informed consent, then it can be saved for research. But it does not happen automatically. There is a whole process of getting consent, signing off on various waivers, and so forth. So it's not automatic. There is a whole process for it. All right, I'm going to stop here and let's take a 5-minute break. And by 2 p.m., my students will come and they will be presenting some very exciting diseases which hopefully have a potential to be fixed in the next few years through the use of stem cells. So we're going to take a little 5-minute break at this point. Thank you so much. So Fatima, I think you can actually get your slides ready.
Would you like me to start sharing now?
Yeah, maybe the first title slide, and
Yeah,
then
yeah.
you can start talking in about 4 minutes.
Okay.
Thanks. going on? Okay, Fatima, I think you can start now. Fatima, and please introduce yourself, all five of you, and then please get started. Thanks. You muted, Fatima.
All right, everyone. First of all, thank you so much for having us here today. My name is Fatima, and with my colleagues Joseph, Casey, Georgina, and Victor, we will try to introduce you some applications of stem cells in the leukemia treatment and how the stem cells are changing the approach for this disease. Here's just an overview of what leukemia is. We're going to just talk about very briefly, and then we're going to jump to what hematopoietic stem cells are, and then we're going to go over the transplant process and some challenges and risks for patients and doctors, and we're going to end up with some success stories and future directions of this disease. So let's just start when the blood formation goes wrong. So leukemia is a blood cancer and is characterized by the formation of abnormal cells in our body. So most of the cases, the patients will go to the doctor when they start feeling some weakness, some shortness breath, and some weakness for low energy, and also the most cases they will start seeing bruises on their skin. And the diagnosis involves not just the physical medical history of the patient, but they will also combine a bone marrow biopsy and some radiology exams, and including blood tests and sometimes the spinal tap. And this is a very complicated process because it will involve this is going to bring to the doctor to know what's the exactly treatment for each patient. So how it starts in the bone marrow. The bone marrow is this soft and spongy tissue that is inside of our bones. And here on the left, we can see a healthy and a leukemia bone marrow. And when that's when it starts to go wrong. In the bone marrow is where all our blood cells are going to start to be produced. And here on the right, we have an example of our normal blood and a leukemia blood, because in leukemia, what happens is the marrow will start producing a lot of abnormal white cells, and these white cells are going to start to overcome the other cells in our body. Here is just some facts for you, an overview of about 67,000 new cases of leukemia are diagnosed in the US every year. And with that, the leukemia accounts for about 3% of all new cases in the whole country, and that puts the disease in the 10th most common cancer in the US. So sadly, half million people in the US are currently living or are in remission of leukemia. So that's a lot of our people that needs treatment, and that's one of the reasons that it's really important to look over this disease. So unfortunately, until today, there is no one single reason and research don't fully understand what's the cause of all type of leukemias, because there are a differentiation of the type of leukemias and the phase of each disease. But they know that there are increase of risk factors that can be involved with developing this disease. For instance, a personal history family with someone that already had disease before, or if the person has been submitted to previous other cancer therapies, some chemical exposure, or genetic factors such as mutations, or even if the patient already had some conditions with some type of virus, for example, Epstein-Barr. And also it's more common in females and smoking patients and with increase of age. So, and that's very important to know because usually it's not just one factor that is involved, it's a combination of all these factors.
Hello, I'm Joseph, and I'm here to cover hematopoietic stem cells, which are the blood system's foundation, and how they are used in leukemia treatment. So, what are hematopoietic stem cells, also known as HSCs? They are stem cells that they're what they do is they differentiate into other blood cells to help replenish cells in the bloodstream. They can turn into red blood cells, white blood cells, platelets. They're multipotent, so they can turn into many different things, not anything, but uh, pretty much everything you can think of that you need for blood. Uh, they're found in the bone marrow and also the peripheral bloodstream, which is the blood that actually goes through your veins and arteries. And they can also be found in the umbilical cord as well. Uh, they replenish all blood cell types by undergoing hematopoiesis, and they spend most of the time dormant, not actively creating new blood cells until uh, some sort of loss of blood cells occurs, in which case they can be stimulated to start undergoing hematopoiesis to create new blood cells. And next slide. So how can HSCs be used to treat leukemia? So in leukemia, patients have abnormal HSCs that produce abnormal white blood cells, as Fatima brought up. Uh, and to treat leukemia, patients go through chemotherapy. But in the process of chemotherapy, all of these HSCs, which are extremely important for the body and maintaining a healthy bloodstream, are mostly destroyed. So patients need a transplant of healthy new HSCs to so that their body can recover and start producing healthy blood again. Uh, HSCs can be either taken from themselves or by a donor uh, to be introduced into the patient so that they can start making healthy blood. And without this stem cell treatment, the patient would not be able to truly recover healthily from the chemotherapy.
Thank you, Joseph. And Fatima, we can go to the next slide. So to prepare for the stem cell transplant process, you first have to prepare the patient's body. As we've learned, leukemia causes abnormal blood cells to grow uncontrollably and crowd out our healthy cells in the bone marrow. So to treat this, patients are going to receive a high-dose chemotherapy and sometimes radiation therapy. And this serves two purposes. First, it destroys as many of the leukemia cells as possible. And second, it creates space in the bone marrow for the new stem cells that will be transplanted. And one way to think about this process is like preparing a garden. First, before planting new seeds, you have to remove the weeds and prepare and clear the soil. So the conditioning treatment helps create the best possible environment for the new stem cells to grow. And although this treatment is very effective, it temporarily removes many of the healthy blood cells and weakens the patient's immune system, which is why the next step is so important. And so we can move to the next slide. So we're going to be moving on to the stem cell and engraftment process, and we're receiving our new stem cells now. And the transplant process is very interesting because it sounds like you're receiving a transplant in the sense of an organ transplant, but there's actually no surgery involved. So once the conditioning treatment is complete, you're ready to receive those donor cells. And the stem cells are infused through an IV, much like a blood transfusion. And after entering the bloodstream, the stem cells travel throughout the body and naturally find their way to the bone marrow. And once they arrive there, they begin the process known as engraftment. And the engraftment is when the donor stem cells settle into the bone marrow and start producing the new blood cells. And these new stem cells generate all the major blood types, including the red blood cells, the white blood cells, the platelets. And in many ways, the transplant is like installing a whole new blood forming system. And so the patient's previously diseased bone marrow is now replaced by a healthy stem cell, by healthy donor stem cells capable of rebuilding the body's blood and immune system. Next slide, please. So the recovery after transplant is very interesting. So engraftment typically occurs within 2 to 4 weeks. The immune system may take much longer, months to recover. And so you do have to monitor your patients very closely for infection. And a major complication is graft versus host disease, which I'll get into a little bit more in a second. So as the transplanted stem cells begin to engraft, they start producing new blood cells. This is when we begin to see those blood counts begin to recover. And for many patients, engraftment occurs within the 2 to 4 week period after transplantation. Although the infusion itself is really quickly, it only takes a couple of hours, the recovery process is much longer. And those patients, again, are closely monitored for infections, complications, and signs that the new stem cells are functioning properly. And the graft versus host disease, which is when the donor immune cells attacks the patient's tissues. This is why finding the closest donor match is so important, and the transplant teams are very careful when they compare the tissue markers called human leukocyte antigens, or HLAs, when they search for donors so that they closely match. Actually, a family friend just received their stem cell transplant for leukemia last October, and they had a very high HLA match, and his transplant team actually searched internationally and found his donor in Europe. This highlights how important, worldwide donor registries are and the best possible match for each patient. Fortunately, he got a very excellent match and has done very well. And you can't keep a good man down with that one. And he actually had a very fascinating thing with his transplant and recovery, is that his blood type is now changed to his donor's blood type. And he's also experienced remission of his psoriasis after his transplant. And the ability of these transplanted cells to completely rebuild the new blood and immune systems is one of the most successful examples of how stem cell therapy works in modern medicine. And so now that we've seen how stem cell transplantation works and how patients recover, our next speaker will discuss donor matching and other considerations in leukemia treatment.
Hello, I'm Georgine, and I will present some of those challenges and risks. As we just learned, one of the major challenges in stem cell transplantation is finding the right donor. Stem cell transplants have the best chance of success when the donor and patient are closely matched. In this case, family members, especially brothers and sisters, are often good matches, but many patients unfortunately do not have a suitable family donor. In those cases, donor registries can help find compatible donors. Thankfully, over the years, these registries have grown, giving more patients the opportunity to find a life-saving match. One example is the National Marrow Donor Program, known as NMDP. Through this registry, people can volunteer to become a potential stem cell donor. And for that reason, as part of our biotechnology program, we volunteer with NMDP to help raise awareness and encourage people to join the registry, because every new donor who joins increases the chance that a patient will find a life-saving match. Next, please. Finding a donor is a major step, but the challenges do not end there. Even after successful transplant, the complications can occur, and the most common, as Casey mentioned, is called graft versus host disease, known as GVHD. And this happens when the donor's immune cells recognize the patient's bodies as different and begin attacking healthy tissues. The For example, the most common areas in this case are skin, where the patient can get rashes or redness, abnormal liver or intestine function, and therefore doctors monitor patients closely for these complications during the time during the weeks after the therapy has been received. Today, several medications can help prevent or manage these. Thanks to these advances, stem cell transplants have been safer and more successful than they were in the past. Next, please. Um, Stem cells have helped many patients achieve long term remission and in some cases a cure. Since before receiving the healthy stem cells, patients often undergo chemotherapy to destroy the leukemia cells and prepare the body for the transplant. During recovery, patients are also carefully monitored and receive treatments to help protect them while their immune system rebuilds. Therefore, when creating a treatment plan, doctors also consider factors such as age and overall health because every patient is different. So, stem cell therapies have to be tailored to each patient's individual needs. Today, stem cell transplantation has transformed leukemia treatment and continues to offer hope to thousands of patients each year. Advances in transplant medicine have improved outcomes and expanded the treatment options for many people who once had very limited choices. Thanks to these advances, stem cell transplantations have become an important tool in the treatment of leukemia. And next, Victor will discuss some of the success stories and exciting future developments in stem cell therapy.
Thank you, Gina. So, hi, my name is Victor, and I'll be wrapping up really quickly. Apart from challenges, let's also look at some of the successes that stem cell treatments have had for leukemia. So, uh, Stem cell transplants do save lives, and they save lives every day. In this regard, there have been more than 1 million hemopoietic stem cell transplants performed worldwide. Many patients do achieve long-term remission, or in fact, end up being like cured after transplantation. Survival rates have continued to rise due to better donor matching, improved infection prevention, better management of graft versus host diseases, and also because of earlier diagnosis and treatment, just due to better systems. Next slide, please. So, thank you. Oh, okay. So we can see here, comparing stem cell treatments versus non-stem cell style treatments, the rates are higher, right? Of especially for these three first that I have listed: AML, acute myeloid leukemia, acute lymphoblastic leukemia, and relapse. Uh, Stem cell treatments have much higher success rates, especially over 5 years, than non-stem cell related ones. There are other diseases or conditions where traditional or targeted therapies are just as, if not more, successful, and stem cells have a much lower emphasis, such as chronic myeloid and chronic lymphocytic leukemia. So next slide. Uh, Thank you. So traditionally, the best outcomes were achieved when patients received stem cells from a fully matched sibling donor. However, unfortunately, only about 25 to 30% of patients have a brother or sister who is a close enough match. So, for many years, patients who could not find matching donors often had limited treatment options. So, this is where we look at the next generation. Some of the major advancements that are being pursued now are umbilical cord transplants. So after a baby is born, the blood remaining in the umbilical cord contains hematopoietic stem cells, and that can be collected and stored in specialized cord blood banks. So these stem cells are more flexible than adult donor cells, as was previously discussed, and they can be used, and they provide expanded transplant opportunities for people that maybe have less donor options, usually like mixed race people or people from underrepresented groups. And then another exciting development is the rise of haploidentical transplantation. In these procedures, stem cells are obtained from a half-matched family member, like a parent, child, or sibling. Advances in transplant medicine and immune-suppressing therapies have dramatically improved the safety and success of these transplants. As a result, nearly every patient today has the potential to find a donor within their own family. So advances like these are helping to make stem cell transplantation much more accessible than ever before. Looking ahead, researchers are exploring ways to expand the cord blood stem cells in laboratory, develop universal donor stem cells, and use gene editing technologies such as CRISPR to further reduce complications and improve outcomes. And yeah, so the ultimate goal is simple, right? It's to ensure that every leukemia patient who needs a stem cell transplant can receive one, regardless of their background, ethnicity, or family circumstances. Thank you.
Great. There's a question on the chat for you guys. The question is the IV infusion only done through the arm, or is the femoral artery ever used since it's larger?
It's typically an IV to the arm or like a PICC line.
Right, because in the interest of time, we will move to group number 2, and that is Alex. Please go ahead and project your presentation.
All right, can everyone see it?
Yes.
Yes.
Wonderful. Hello, everyone. My name is Alex, and I'm joined today by our Lee, Kyle, Owen, and Wenke. Today, we'll be discussing Alzheimer's disease and how current stem cell research is expanding our understanding and treatment of the condition. So first, let's talk a little bit about what Alzheimer's disease is. So it's considered a progressive neurodegenerative disorder, meaning that brain tissue and function degrades over time as the disease progresses. The most prominent symptoms are a loss of memory, a loss of the ability to think properly, and like I said, the degradation of brain tissue as the entire organ begins to atrophy in response to the condition. Biologically, there are a couple of indicators that the symptoms in question are due to Alzheimer's as compared to other dementias. One is the buildup of amyloid beta plaques in the brain, and the second is of neurofibrillary tangles within the neurons. These buildups occur primarily in the regions that are responsible for things like memory, language, and judgment. So now let's talk a little bit about what's going on biologically in the brain suffering from Alzheimer's disease. These diagrams go into depth about what's going on at the cellular level, but I'll break it down for you into less strict scientific terms. So on the left here, we're seeing the creation of amyloid beta proteins by neuron cells, and this releases them into the brain. Amyloid beta peptide proteins are actually natural products, but in Alzheimer's, these proteins clump together into what are called plaques, and these clumps of gunk prevent the neurons from properly sending signals. And then on the right, we see a comparison of healthy neurons and neurons affected by Alzheimer's. So within neurons, and neurons are the main cells in the brain responsible for sending the signals that control your body and mind. So within these cells, there are these things called microtubules. And microtubules are basically just these long fibers that provide structural support. And by providing structural support, it allows them to send signals more efficiently. In patients with Alzheimer's, these fibers end up tangling together, tearing apart the microtubules and releasing something called tau proteins. So basically, what you're getting in Alzheimer's is this accumulation of plaques and tangles in large numbers. And that creates an environment in the brain where the necessary signals to function are disrupted. And this dysfunctional ability to send signals in the brain directly influences the cognitive decline of Alzheimer's patients.
Okay, so why do we care about Alzheimer's? How does Alzheimer's affect us? In the United States, the highest concentration of Alzheimer's are in the eastern and the southeastern United States, including our state. Moving on to the next slide. If we zoom in on the numbers, here in Ventura County, per 100 older adults, nearly 12 of them are living with Alzheimer's, and this number will only increase. Let's put a pin in that. Nationwide, 1 in 9 older adults live with Alzheimer's, which further overwhelms the healthcare infrastructure as well as the labor market. Importantly, 2/3 of the cases are women. Well, this translates to a loss in female expertise and leadership in the workforce. Moving on to the next slide, why will the numbers only increase from here? Well, as we enjoy higher standards of living, we also observe an increased life expectancy as well as lowered Alzheimer's mortality rate. This means more people can live longer even with an Alzheimer's diagnosis. On the flip side of the coin, our lifestyle choices can also lead to other illnesses that co-occur with Alzheimer's, like hypertension, etc. This can exacerbate Alzheimer cases. The other important factor that may not be intuitive is actually the climate change, which causes neuron inflammation that makes people more vulnerable to neurological injury. This is because a warmer climate exposes more environmental toxins, which puts more stress on neurons, and it triggers harmful immune responses that lead to tau tangles as well as amyloid plaques. I will hand it off to the next person. Thank you.
Thank you, Winky. I'm Owen, and I'm going to talk about some of the current treatments used for Alzheimer's today. So these current treatments, they mainly focus on slowing the progression of the disease, as well as managing cognitive and behavioral symptoms. But most importantly, the focus is on maintaining a patient's quality of life and independence. So as of yet, there is no cure to Alzheimer's disease, so the current treatments mainly focus on mitigation and prevention. But early diagnosis of Alzheimer's disease has been proven to be critical because some of the newer therapies work best in the early stages of Alzheimer's. Next slide, please. So cholinesterase inhibitors, such as donepezil and galantamine, are used to treat symptoms of Alzheimer's. So they've been shown to improve memory and cognition in patients by increasing the availability of a key neurotransmitter, acetylcholine. Memantine is an MDMA receptor agonist, and it's used to help regulate brain signaling and has been shown to improve cognition and daily functioning. But it's important to note that these benefits are usually modest in patients with Alzheimer's, and these drugs, they don't stop the underlying disease. They're just used in patients to help mitigate symptoms, and they're most effectively used when the disease is diagnosed early on. Next slide, please. So some disease-modifying therapies are donanemab and lecanemab. They're monoclonal antibody therapies, and they're used to help target and then clear amyloid beta plaque in the brain. They're delivered intravenously, and they've been shown to help slow the cognitive decline that's often associated with Alzheimer's disease. But again, these therapies work best in patients with early onset Alzheimer's, and they're also not a cure. They often require periodic monitoring in patients with MRI scans also. So a common theme in these approaches is that none of these therapies actually stop Alzheimer's, right? They're used to treat symptoms and slow the progression of the disease. But some alternative approaches have been promising, such as stem cell therapies.
All right, so thank you, Owen. I'll be taking over the stem cell therapies and where they are in research right now. This trial was sponsored by, or is sponsored by Hope Biosciences, and is going to be conducted at UT Health in Texas. And the goal of this trial is to understand whether stem cell therapy reduces brain inflammation. Inflammation in the brain can be involved in adults with dementia and also in adults with Alzheimer's. And so this trial right now is in phase 1B, and it works by taking a fat sample from the patients at a doctor's office, and it takes cells from those and processes them into stem cells. And they would be receiving 4 infusions of stem cells through a vein in the arm over 12 weeks. And they would be visiting the clinic every 2 to 4 weeks for the first 4 months, and then every 1 to 2 months for 8 months for checkups and tests. And it's recruiting as of right now, and it's looking for 12 individuals. And this trial is based on prior animal research models that shows positive effects in reducing the amyloid plaques in mice and other animals. And so while they're recruiting as of now, they are hopeful for positive results. Next slide, please. So this other stem cell therapy was based on the human umbilical cord blood cells and turning them into mesenchymal stem cells, which basically those are multipotent adult stem cells, and they're found in bone marrow, adipose fat, and umbilical cord blood, and they differentiate into bone, cartilage, and fat cells. Basically, this stem cell therapy used one single infusion, and they studied a low dose and a high dose. And the results of this study found that there were no serious side effects like fever, which is commonly found in stem cell therapy treatments. But the efficacy still remains mostly unknown, and further trials are necessary to better understand how these therapies work. And some of their reasonings for why the trial maybe didn't replicate animal studies as well could be the imaging systems and how they were not able to be as sensitive to detecting soluble amyloid plaques. And also, the environment, the Alzheimer's disease environment, differs largely between humans and mouse models, and that could be another reason for these results. But these trials are basically building evidence and improving further stem cell therapies. And scientists are at large hopeful that we will find some stem cell therapy that does work to treat Alzheimer's disease.
So this is a novel option made available by the bioengineered injection instead of the more common way, which is an invasive surgery that is both painful and repeated. This involves a bioengineered tool called an Ommaya reservoir, which is a minimally invasive implanted reservoir under the scalp, which enables even and targeted stem cell infusion into the brain. The photos are also sponsored by the MD Anderson, which is a stem cell research powerhouse.
So we can see the potential stem cells have in terms of Alzheimer's disease, but where are we currently at with them? Well, right now it's mainly in the research and clinical trial phases. There's no real regenerative medicine therapies for Alzheimer's at this current moment. So unless you're participating in a clinical trial, using stem cells to treat the Alzheimer's disease is not currently available. So when it comes to research, uh, primarily, this involves use of animal models, specifically mice, that will model some form or fashion of Alzheimer's. And then different types of stem cells and different modes of implementation are being used in these animal models to study how they can affect the disease. And a lot of the findings are very promising. There's a lot of alleviation of symptoms. So this is increased learning and memory, reduced neuroinflammation and plaques, and also increased density for neurons and synapses. And then stem cells can also be used to model the disease. So not only can it be used as a therapeutic, but it can also be used for therapeutics to be studied on it. So this is where organoids come into play, and these are small um, tissue structures that resemble the development of organs in your own body. So in this case, you can use these stem cells, grow them up into organoids. In this case, it'll be a brain-like structure, and this will resemble human brains. So this way, stem cells can model Alzheimer's, and therapeutics can be tested on these stem cells so we can get a more realistic model to humans as opposed to using mice. And now in the clinical trials, like it was mentioned before, the stem cell therapies that have progressed to the point of clinical trials are still phase one and phase two, which means they're strictly testing for safety and efficacy. So this is where we're at the moment, but there's a lot of promise here seen in the research for the animal trials and then in the clinical trials There has been some promising results and still some things to figure out in terms of maybe a lack of results, but there hasn't been any significant negative side effects. Next slide, please. So we can see that there's real, genuine therapeutic potential for Alzheimer's disease using stem cells. And like Owen touched on before, currently the treatments are simply to slow symptom progression, but stem cells actually can alleviate symptoms and work towards more of a cure rather than just a treatment or a halting. So in the future, as we progress with stem cells and work with them in terms of Alzheimer's disease therapies, that will involve trying to target specific symptoms of the disease. It could be combining stem cells with other currently available therapeutics. So it could be with possible monoclonal antibodies. It could be trying to use a current neuroinflammatory along with stem cells to try and eradicate any symptoms. And then disease modeling, like I mentioned, with the organoids, that will continue to progress with stem cell research. And then finally, one aspect that's still being worked on and will continue to be worked on is using the stem cells that already exist within your brain and using sort of therapeutics or something to stimulate those in order to help with the Alzheimer's disease progression. And so there's still many years to go in terms of research and clinical trials before any of this can become marketable. But stem cells show true potential in the treatment for Alzheimer's disease. And this is a new, accessible, and effective strategy that over time can be a potential cure for this disease. And next slide, please. I wanted to just close out on this image that I found super interesting. And this was just stem cells that were, this is in a mice or a mouse, and these stem cells are in the green, and they're differentiating into these red microglia-like cells, which are actually reducing the plaques in purple. So I thought this was a cool visual to sort of end our presentation on, so you can get an idea of what's going on inside the research and how this can really develop in vivo in humans in the future.
Thank you so much, Group 2. Yes, that was great. I think the potential is strong. It's very optimistic. Hopefully, in the next few years, we can see this in practice. So there's no questions at this point. We will quickly move to Group 3, and that is Tirth, Boston, and Atharola. Atharola, please start projecting.
Hello, everyone. Good afternoon, everyone. Today, my group, Ataula, Agustin, and I, Pete, are going to talk about multiple sclerosis or MS in a way that is simple and clear to connect with. So, when people talk about MS, they may know it as something to do with brain or nerves, but they may only know exactly what is happening inside the body. At its core, MS is a condition where the immune system, which normally protects us, starts attacking the parts of nervous systems by mistake. The nervous system is how brain communicates with the rest of the body. Every movement we made, every sensation we feel, and even things like balance, vision, coordination depends on those messages traveling smoothly. Next slide, please. In MS, the protective covering around nerves, called myelin, becomes damaged. A simple way to imagine this is like a phone charger with worn out insulation. The wires may still be there, but the signal or power does not travel as well as it should. That is why MS can look very different from person to person. One person may mostly experience fatigue, another may have vision problems, numbness, weakness, or trouble with balance. Some people have symptoms that come and go, while others may notice symptoms slowly getting worse over time. So one important thing to remember that is that MS is not same experience for everyone. Next slide, please. Now, when we ask about causes of MS, the honest answer is there's no single cause. It is not something that is caused by one habit, one infection, or choice made. Instead, researchers believe MS develops over several factors come together. So part of it involves immune system becoming confused and attacking myelin. Part of it may involve genetics. Maybe some people may be born with a slightly higher risk, but MS is not directly passed down in a simple way, like eye color. Environment also seems to be the matter here. For example, low sunlight exposure and lower vitamin D levels may play a role, which is one reason seen in MS more often in certain regions of the world. There is also growing research on infection, especially Epstein-Barr virus or EBV, where people are exposed to EBV in their lifetime. But studies suggest it may be one important trigger in people who are already at risk. So hormones may also influence risk since MS is most common in women. And new research is even looking at gut microbiome, meaning the bacteria in our digestive system and how it may affect immune function. So the best way to understand MS is not a disease with one clear cause, but a combination of immune system changes, genetic risk, environmental factors, viral expose, or other influences. So this is what makes MS complex. But it is also why research is moving in a promising direction. So with that foundation, we can look at how MS is diagnosed, what symptoms may people experience, and what treatments are available today. Ataula will continue.
All right, hello everyone. My name is Ataullah Muhammad, and I will be talking about the diagnosis and the treatments that are available for MS patient currently in today, and how the stem cells are being used to find a cure for MS. So generally, when we help in diagnosing multiple sclerosis, it's just not something that's just done by a single test, or a single test could show you the confirmation for MS to be diagnosed So we use like four methods. That's MRI imaging. That's one of the most trusted method and one of the most gold standard method that we use to find white plaques. That gives a confirmatory test whether the patient is suffering from MS or not. The other methods are CFS analysis, where there's a lumbar puncture where a needle is punctured into the spine and spinal fluid is drained, and the spinal fluid is then looked under microscope for oligo IgG bands, which are one of the antibodies, and in 90% of the cases, you find these bodies. The other one is the nerve signal, that's the EEG, where a helmet-like cathodes are placed, and your nerve signal speed is measured, and anything slowing your nerve speed might lead to suspecting MS. Other one is the clinical history. Because MS can happen during any time. They're usually, to get a confirmatory result, we do 2 episodes or 2 periodic episodes of multiple sclerosis. So what we do is whenever there's a single episode, it's not a confirmatory test, but one single sorry for that. So one episode along with MRI imaging leads to the diagnosis and would confirm the diagnosis of MS in a patient. So how you decide whether it's a confirmation or not is done by a criteria that was revamped in 2017, known as the McDonald criteria. In McDonald criteria, there are 2 ways you can do. There's one is in space, dissemination in space, and one is dissemination in time. So dissemination in space means if in MRI scanning you find any plaques in 2 or more regions of the CNS system, that includes your periventricle, cortical, or even your spinal cord, it confirms the diagnosis. And dissemination in time means finding new lesions over time, or follow-ups, or regular checkups, leading to new symptoms, new plaques showing up in the MRI images, lead to dissemination in time. So this also leads to confirmation of MS. Next. So these are the MRI scans over here, and you can see these are at different. There's a post-injection of flare injection that's injected, and all the all the white plaques you see in the image A, B, C, and D are different signals, where there's more highlight, more emission of the white plaques As you can see in the C level, it's more well prominent, and you can see very well how the white plaques are, which indicates the formation, the plaques that indicates the confirmation of multiple sclerosis in the patients. So next we're going to talk about how this disease progression. It's not something, MS is not something that's just happens once and it's gone. It's a debilitating disease that over time gets worse, and the quality of life of the patients reduced significantly over time. So generally the symptom, there's three phases which we can say how disease progresses. There's a relapsing phase where there might be episodes at intervals of time, and there's no set interval between when it can happen. It can be sometimes it can take weeks, or sometimes the episodes can range from weeks to years in happening. And after every episode, there's a risk that the patient might not recover fully. So there might be partial recovery, but full recovery, or sometimes in some cases, full recovery. And then there's a progressive phase. In this case, even without relapsing, there's a progressive degeneration of the musculoskeletal functions, and over time there might be no relapses, no sign of any new episodes, but slowly and steadily the condition of the patient worsens over time. And in some cases, which is less than 1% of the cases, there's significant disability, and there's even cognitive and autonomic involvement where people are not oriented to where they are. It might lead to dementia, vision problems, muscle weakness, and even paralysis or bedridden over time. So you can see over here how the common symptoms are in most of the MS patients. Most of the MS patients report that they have 80% fatigue, 60% of them have balance issues, and how cognitive issues, like 50% of the cases are cognitive issues. And over time, you can see how other muscle weakness, vision problems, and mood changes over time. And these are very high. These symptoms altogether combines you, or anyone suffering from these symptoms might require a test for MS, but these are generally over long time or over duration where the symptoms might worsen. And how do we know at what progression MS patient is? So this is one of the scales that was in McDonald's criteria that gives a disability status to the patients, and it shows how it is. As you can see over here, 0 to 1.5 or 0 to 2 might be a normal patient. Means they are functioning normally. They can do pretty good function. They can walk around. No disability is seen. In mild cases, you can see there might be minimal disability, like there might be vision problems, there might be fatigue over time, and they might not need full rest over time. But in moderate, here's where you can see how it starts affecting the patients over time, like how like walking only 500 meters without any aid. But after 500 meters, there's the patients get pretty tired, and over time you can see how at 5.5 in moderate conditions, the patient can't even walk 100 meters without any aid and can't do any full day activities. In severe and very severe cases, they might require a cane to carry on, sometimes a wheelchair, and in very severe cases, they're bedbound and they're very helpless and cannot, and they can only communicate and eat and they can eat, but in very severe, at the last point, there is total helplessness. They can't eat, swallow, and they can't even do basic functions. So let's move on to a pretty bright topic after this, because the symptoms and everything else seems scary. So we can see MS has come a long way from last 20 years. People, before, it used to be a disease that there was no treatment for, and the treatment were very minimal, and they didn't have any how to work on how to have a better quality of life or how to give a better treatment to the patients. So now we are moving on to better treatment options. One of the treatment options that we have is the disease modifying therapies. There are 20 FDA approved drugs. All of these are monoclonal antibody drugs. Some of them are beta interferons. but one of the most prominent drug that's being used right now and is the first child of drug for physicians and doctors around the world is the Ocrevus or ocrelizumab. It's a monoclonal antibody, and it has shown great results, and it has reduced around 70% relapsing remitting multiple sclerosis, meaning it after the first episode, the second giving this or taking this over time has reduced the number of episodes occurring in multiple sclerotic patients. Okay. So, and other methods are mostly the methods of symptomatic management that we do, that is relapse management, where high dose IV corticosteroids are given, where plasma exchange is given in severe cases, and rehabilitation therapy is given, mostly to reduce the episodes and to reduce more damage to the body, reduce the damage to the nervous system. Third one is how you manage the symptom conditions. That some of the things are like people can use diet and gentle exercises. For fatigue, they are given amantadine, and physical and occupational therapy is pretty good for patients to have like better muscle response, better mobility. And for psychological support, CBT, mindfulness, yoga, and meditation are also performed to have to keep in check the mental status of the patients. So acute relapses, as I have told you, in cases of relapsing remitting, which occurs in 85% of the patient, most of the patients have high dose. How are they treated whenever they're suffering from an episode? What you do is you give high dose corticosteroids to them, which is the first line of treatment. As soon as someone's suffering from episode, the first thing they're given is a high dose corticosteroid to reduce the inflammation and to reduce the immune response of the body, so that they have a speedy recovery, and also to reduce the long-term effects that the episode might have. In other cases, it might be plasma exchange, which is also known as plasmapheresis, in which the immune antibodies from the blood are removed, and when the corticosteroids are not effective on the patients in severe relapses, this is one of the methods that's used to help rescue the patients in the severe cases. So how does stem cell come into place for multiple sclerosis? So here you can see that one of the methods that is to reset the immune system. And how do you reset the immune system? One of the methods is the hematopoietic stem cell transplantation, as discussed in the earlier presentations by my classmates. In this stem cell transplantation, patients' own blood stems are taken, this harvested, chemotherapy is done to the blood cells and wipes out the old and the faulty immune cells. And then the stem cells are harvested and then again reinfused into the healthy immune system. For whom this is best for? This is best for younger individuals, possibly from 20 to 40 years of age, and people who are suffering from RRMS early in the disease or in like severe or in the early phase of the disease. It has shown pretty good response. And one of the trials that's going on with HSC transplantation for MS is the Beat MS trials. They have shown promising results in phase 1 and phase 2. And phase 3 is considering with it's doing a comparison between the DMTs that I have talked about previously and how the HSC are, and trying to find which is better efficient in this system. The other method is the mesenchymal stem cell system. In this, the MSCs are harvested from the patient from bone marrow for fat, and it's grown and cultured in the lab. And then these are stimulated to release chemicals that lowers the inflammation over time. So what's the goal of MSCs? It's to stimulate remyelination and help in regrowing protective myelin so that it's well tolerated, so that it's well managed, and to maintain the integrity of myelin structure of the neurons. In phase 1, so these are pretty early in stage right now. Some of them are phase 1 and 2 trials, which are showing results as safe and well tolerated, but there's still a larger population and larger cohort needed to get the approval for a phase 3. One thing about stem cells is they don't come without their risks and considerations. One of the risks, especially in autoimmune diseases regarding stem cell treatment, is that it can alter the immune system of the patients. And if this leads wiping out the immune system, it carries high risk. It can alter the immune system. It can make the body more susceptible to more autoimmune systems. So it comes with risk. It's caveats, but generally, in most of the cases, it's shown to have a better result and better efficacy. And it is not still, it's not a universal cure, and it's pretty highly selective, and it's still in the early stages. Next, it talks about. Next, I'm going to just talk about next time. We're just going to talk about how patients are going to, how you can carry on, and how quality of life should be maintained, and how you can help with patients with multiple sclerosis. First and foremost is the physical health. In patients with MS, it's mostly due to it being a musculoskeletal disorder. Patients generally get fatigued easily, so they need a gentle cardiovascular exercise, stretching, yoga, swimming, and constantly trying to do more training. It's also shown that light resistant training also helps with building muscles, and this can help with having a better quality of life in later stages of the disease. One of the other is the mental well-being, where I have talked about previously how CBT and how mindfulness can help reduce depression and anxiety, which affects in 50% of them as patients. So psychological support is very important in patients with MS. Multidisciplinary care, you have to do follow-ups, you have to keep checking up on the patients and coordinating with all the specialists and social workers, forming support networks, peer groups, to talk about disease, to help them get over things and help them provide so they don't feel more lonely and isolated and feel that they are not alone in this fight. One second. So this is some of the topics in the future of MS and how we are gonna how there's treatments and how the cures are being worked on. One of those is a remyelination therapy, in which drugs like LINGO-1 antibody, opsinumab aim to repair the damaged myelin. And these are one of the first potential cure pathway. We still don't have a cure for it, so this is the treatment. So these are one of the methods that we are working on. The other method is the neuroprotection, where rather than remyelinating the neurons, they're working on protecting the neurons. The drugs that are currently available in the market are simvastatin, ibudiblast, that helps in slowing the degeneration of the myelin sheath. And the third one is the stem cell therapy. And all of these in detail, my classmate Augustine is going to talk about in the next slide.
Sorry to interrupt, but I think you'll have to wrap up pretty quickly because we are really nearing the end here.
All right, yeah, just like some slides left, so it's all done.
I'll keep it brief. Thank you. I'm Augustine. I'll be talking about stem cells versus the versus current treatments. So it's helpful to understand why researchers want to study and focus on stem cells in the first place. So today there are more than 20 FDA-approved treatments, and a lot of them are really effective at reducing relapses and slowing down disease progression. But stem cells offer a unique take on therapy because they may repair damage, restore function, and they have potential long-lasting benefit that aim to rebuild the damaged myelin. Whereas in traditional medication, focuses on slowing down progression, reducing inflammation, preventing future attacks, and they require ongoing treatment to manage the disease. Next slide. So what are researchers seeing so far? Whenever we hear about new treatment, the most important question is always, is it actually helping patients? And researchers have actually seen pretty encouraging results. And so a lot of them have seen long-term remission where many of the patients experience years without new relapses. There's stable MRI findings where the MRI scans often show little to no disease activity, and there's improved daily living where the patients report improvements in quality of life. And so, as he mentioned a little bit earlier, the strongest results are early in MS in younger patients who have been diagnosed early on. And so, the benefits appear strongest in this population of a highly active disease. And so, the safety is still under investigation, and researchers are still studying the long-term safety of stem cell treatment. Next slide. And so, looking towards the future of stem cell therapy and multiple sclerosis, it's a promising future. Um, right now, the treatments are focusing on controlling the immune system and preventing further damage, whereas in future therapies might go beyond that goal and focus on repairing the nervous system. And so they're looking at restoring myelin, which is the protective coating around the nerve fibers damaged in MS. And they're hoping that future stem cell-based therapies could help the body rebuild this protective layer and improve communication between the nerves. And so they're also looking at protecting the nerve cells themselves. So as multiple sclerosis progresses, damage can accumulate over time, which leads to permanent disability. So the scientists are investigating whether stem cells can help preserve these cells and maintain neurological function for longer periods. And so they're also working on to making these stem cell therapies more accessible. Currently, these procedures are very invasive and can only be performed in specialized centers, whereas in future approaches may be less invasive and still provide meaningful benefits to the patients. But ultimately, the real goal is much bigger than just reducing relapses or slowing progression. It's about helping people maintain their independence, preserve mobility, and continue participating in their daily activities that matter most to them. Next slide. And so, just some key takeaways is that multiple sclerosis is a complex autoimmune central nervous system disease. And so, genetics, environment, and immune factors all interact to cause it. Diagnosis relies on the McDonald criteria using MRIs, clinical history, and analysis. Average delay is about 5 years. And so, there's no cure that exists right now. But like I said, there are over 20 FDA approved treatments that help slow down progression. But stem cell therapies provide an encouraging outlook, and hopefully, the future we can find better cell-based therapies to help combat this degenerative disease. Thank you.
Thank you so much. That's quite optimistic, but I'm just going to read a couple of questions here. So, at least with regard to your presentation, there's a question: is there a trial using stem cells to prevent further deterioration from MS?
So all the trials that are on MS are mostly focused on autoimmune diseases right now. So most of the trials that I have said before, like neuroprotection and remyelination, they're still in phase 1 and haven't been moved on towards lab. But the only thing that's the phase 3, that's the Beat MS trials, that is the hematopoietic stem cell transplantation. That's in phase 3 right now.
Got it. And the follow-up question is, where are the clinical trials being held? Do you have any idea?
Um, uh, I don't know about the ones that are being held, but one of the trials that have been held is in, I think it's in UK. Some of the trials were held for the Beat MS trials, I think, but I'm not currently sure about the location of it. But
And
I
this
can,
this is a question for all of you, all the presenters. I'm going to read the question. Did you have an opportunity to interview patients with the diseases that you reported on? And if so, did they understand the promise of stem cell therapy for the future, if not for them? This is for all of you.
I did interview my friend's dad, who has had a stem cell transplant for acute myeloid leukemia. And that was part of the inspiration for choosing the leukemia topic.
Great. Anybody else?
So I had interactions during my internship in undergrad with MS patients and people with autoimmune diseases. And when I talk to stem cell about them, it's pretty early in the phase, and I haven't had much with the patients who are in stem cell, but with regarding my disease, people are talking about how DMTs has improved over time. And I have seen how the older patients wish that they could have had the treatments much earlier, or the developments that we are seeing in DMTs right now much earlier than they could have seen. They could have seen it during their times when they first discovered where they were suffering from MS, and that's what they're hoping for with the promise of stem cell and remyelination and neuroprotection, that they could have a better quality of life later in the stage of life. So they wish that we can move more forward with these treatments.
I think the big questions for the patients is, are they pretty optimistic and do they have faith in the stem cell therapies? I think that's a big question.
Yeah, so to answer,
Absolutely.
yes, I think, sorry, go ahead.
He had absolute faith in the stem cell therapy. He had his transplant at City of Hope, and I found that he was very confident in understanding the science behind it with the way they explained it, and they were able to because my friend was asking me about it from my perspective, and he seemed to know the science pretty well from the patient perspective, and felt really confident in understanding that, and seemed to really feel like that was the best step forward as the next step in his treatment. And I think that was probably, and now he's even more happy because he's done really well and hasn't had a lot of complications like graft versus host, that kind of thing.
Thank you. There's one question here. Since the donor match needs to be close, how can the blood types change? Not too sure I understand the question, Bonnie, but I'm going to read the next question. Could you briefly describe the process of obtaining stem cells from donors? Any one of you? From the entire group, entire class here. Anyone would like to answer? How do you obtain stem cells from donors?
So in the case of blood transplantations, there are two types of transplant that they can obtain. 90% for blood cancer treatments are obtained through stem cell. It's like a normal stem cell donation, or 10% are usually from bone marrow. So the stem cells come from these two sources.
But then you can get PBCs, right? The peripheral blood cells.
Yeah.
Also. So
yeah.
you don't have to go directly into like maybe a spinal tap or a bone marrow, but like a thigh, but you could do the PBCs also.
Yeah.
All right. I know we are way past. There's another question. I'm going to read it. My friend had a stem cell transplant from City of Hope and is mad. And he's mad. His golf game is so off. He talked with his donor who doesn't play and hate golf. There you go. It's a comment. Yeah, what can we do, Julie? Better to be mobile and not play golf than not be mobile at all. That's my 2 cents. All right, I think we're going to end here. We are 10 minutes over. So thank you so much, everyone. And I really appreciate everyone for sitting through the 2 hours. Thanks for your patience, and kudos to all the 13 students who spent quite a bit of time. You can see from their quality of presentations, very thorough, very professional, very well done. And these students will be moving on to wonderful sites within California in about 3 months to start their stem cell internships, which will be commencing on September 1. So please wish them the best of luck. And when you see them next year, they'll become very skilled stem cell researchers. So thank you so much, everyone. And thanks to the students. And thanks so much to to Ollie. I really appreciate your time and your enthusiasm. Please take care.
Thank you, everyone.
Thank you.
Bye-bye.